Good afternoon. For our last session, I think the quote is maybe the most important of all of them. It's by my colleague at Columbia, Dixon Despommier. Trust science, not scientists. Today we're going to talk about some of the good uses we can put viruses to, viral gene therapy. And this is going to take advantage of everything you have learned since January, the previous 24 lectures. It's going to draw a little bit from a lot of the basic virology that we have talked about. And we're going to be talking about the use of virus vectors to do a variety of things, including gene therapy, that is, to deliver a gene to patients who lack it or have an altered version of it, for example. We're going to talk about using virus vectors to deliver antigens in the form of viral vaccines. We're going to talk about using viruses to treat cancers, the field of viral oncotherapy. Now, of course, in the end, we use viral vectors all the time in the research laboratory. But today, what we're going to focus on is their use, their clinical use, how you can use them to make people better. Now, the one underlying observation that's common to everything that we're going to talk about today is the ability to manipulate the viral genome. This was not possible before the 1970s when recombinant DNA technologies were developed. And in particular, it depends on having infectious DNA clones of viral genomes. We talked about this a while ago, but let me remind you, it's shown here for poliovirus, that you can take any viral genome, whether it be DNA or RNA, you can make a DNA copy, put it into a plasmid, which is a bacterial vector that can be propagated in bacteria. The DNA can then be modified. You can add genes, you can take away genes, and that's what we're going to talk about today. So this is a brand new field. We could not do viral gene therapy before the 1970s. It's built on a huge amount of fundamental research, not only on recombinant DNA technology, but on the viruses themselves, so we understand what we can do and what we can't do. Remember, with poliovirus, we can take the RNA out and make a DNA copy of it, take that DNA and put it into cultured cells, and out comes virus. And so this can be done with every virus now that we work with. And for the viruses that we'll talk about today that are used for viral gene therapy, this technology enables it. Now, the reason I'm standing here before you today is because of this experiment. This was an experiment I did as a postdoc in 1980-ish. And it was really my main work as a postdoc. And I'd like to say it got me my job, because without it, I might not have been able to come here. So thank you, infectious poliovirus DNA. Let's talk about some of the viruses that are used for viral gene therapy applications. One of them is adenovirus, which we talked about quite a bit in this course, a large DNA-containing virus built with icosahedral symmetry, the structure shown at the top with the very characteristic fibers at each five-fold axis of symmetry. And down at the bottom, the DNA genome. And so after taking this course, something just pops into my head. I spoke to a reporter today, and I was talking to her about viral DNA and RNA. And she said, can you just explain what this DNA or RNA means to the virus? So hopefully one of my goals in teaching you this course is that you'll look at a virus in the future and know exactly what the DNA or RNA means for the virus. I almost said to her, you should take my virology course, but I think it's probably more time than she's willing to spend on something peripheral that she thinks is peripheral to her life. The genome of adenovirus, it's a double-stranded DNA molecule, and this means a lot to the virus, right? It contains the coding region for all the proteins that it needs to replicate, including to build new virus particles, but also to orchestrate the cell so that it can replicate. About a 35 kilobase double-stranded DNA genome with proteins encoded in mRNAs on both strands. We've talked about some of these proteins in this course. Now, adenovirus vectors are used for some gene therapy applications. They have a lot of advantages. They efficiently infect post-mitotic cells. That means cells that are no longer dividing. Remember, adenovirus has ways to kick cells into mitosis, but even before that, they can get into a number of cells very efficiently. They have very rapid onset of gene expression. The genome is episomal. This is a word we've used a bit in this course. It remains separate from the chromosome. So there's little risk of insertional mutagenesis. We'll see in a bit that this is a problem with some gene therapy vectors. Has reasonable capacity up to 37 kilobases, which is a good size. If you'd like to put other things in, that would give you an entire gene if you'd like it with introns, but you don't even need to have genes. You can have cDNA copies of genes, and that would be great. It's easy to make pure concentrated preps, so this is important. You can develop a great vector for gene therapy, but if there aren't techniques to grow it up in good quantity, it's not very good use because you need a lot at some point to put that into people. There are a lot of serotypes, human serotypes and animal serotypes as well. Why is that important? Well, as soon as you give some a vector, you're going to make an antibody response or an immune response to it. So if you need to go back and give them another dose, you have to use a different serotype. Otherwise, you can't do it. And that's part of the drawback of adenovirus. You get a really good immune response to infection, which clears it pretty quickly. So you don't have a lot of time to do your gene therapy. You may have to go back and give multiple doses. Let's look at a little bit at the history of adenovirus vectors to show you how things have proceeded, what kinds of modifications are made to the genome. We have the so-called first generation vectors, the first ones that were developed. They had the E1 and E3 genes. These are early region 1 and 3 genes deleted. So here at the bottom is a schematic of the adenovirus DNA, 2 inverted terminal repeats at either end. Remember Psi packaging sequence here at the left end. The E1 region encodes the T antigens of adenovirus. Remember, those are the proteins that bind RB and P53. And 3 encodes immunomodulatory proteins, which we have referred to in this course. It's not essential for replication. So these were taken out to give some space to put foreign genes in, because you can't add very much to the adenovirus genome without taking something out. Second generation vectors, in addition, they included deletions in the E2 or E4 regions. We'll go back to that original adenovirus genome diagram to see where these are located. So here we have E2 and E4. Again, these were regions that people found were not essential for replication. This is all based on fundamental research, people looking at these genes and asking what do they do, and a side result is that sometimes you find they're not needed, so you can make more space for the transgene by deleting them. So there's the second generation vector. And that brings us to the third generation vector, which is about as far as you can get with these because we have removed everything. We call these gutless vectors. All the genes except the 2 ITRs and the packaging sequences are gone. So here is a schematic of such a genome. If you want to actually make virus with such a vector, you have to put something in. That's called stuffer DNA. You have the 2 ends, the inverted terminal repeats, in the Psi sequence. So to look at how this relates to the genome, at the bottom is the double-stranded adenovirus genome. You've deleted everything so that all you're left with is shown in the middle here. You have 2 inverted terminal repeats, and then in the middle you have the packaging sequences, and here we've left the promoter in, but as you'll see, you could change that as well. So this is a gutless vector. It encodes no viral proteins. So how do you get it to replicate? You need to have a helper virus that you mix with this, and that will package the vector so that you can make virus particles. Now that helper virus has to be defective on its own because you don't want to give people a therapeutic adenovirus which has fully infectious helper virus in it. So the helper itself is E1 deleted. E1 was deleted in the first generation of vectors. E1 is essential for replication in cell culture, but what you can do if you delete E1, you can provide E1 in trans in a cell line. You can make a cell line that makes only the E1 proteins, and then you can grow vectors in that cell line that are deleted for E1. You can do that in principle for any essential gene. The helper virus that you use to package this gutless adenovirus vector is deleted for E1 itself, because you don't want to have a fully infectious virus in the vector virus preparation. So let's take a look at how that's done. This is very clever engineering. The helper adenovirus is a normal full-length adenovirus, except that it has LOX-P sites flanking the packaging sequence, the size sequence. So LOX-P sites are part of a system called CRE-LOX, which involves a recognition site, which is the LOX sequence shown in the red triangles, and a CRE endonuclease, which cuts specifically at the LOX-P site. And so if you surround a sequence with LOX-P sites, the CRE endonuclease will cut it out. So what you do here is, this is the helper adenovirus shown here, the full-length DNA. You have the size sequence with LOX-P in it, and you then can infect CRE-producing cells. This helper is also deleted for E1, so the helper will not replicate on its own. You have to delete E1 in addition, because if this virus accidentally got into a person which doesn't have CRE, it could replicate, so you take out E1. And that's not shown here, but there's a deletion in E1. And then you take that helper virus, you can propagate that helper virus in cells that have E1. And then you take it, you infect cells that are CRE positive, E1 positive, and then you transfect those cells with vector DNA. So we have our gutless adenovirus missing everything except the ITRs and the packaging sequence. You put your gene of interest in it. You take that DNA and you put it into the cells that are infected with the helper. The helper virus will replicate, provide capsid proteins to the vector DNA. Because the helper because these cells are E1 producing, that'll allow the helper virus to replicate. But the cells also have CRE endonuclease. It will cut the size sequence from the helper, so the helper will not be packaged. Right? The only thing that will be packaged is the vector DNA, which has the packaging sequences. So this is a really clever way of having a helper virus provide capsid proteins. And the helper virus will not be packaged because it's got LOX-P sites surrounding the packaging sequence, and those will be cut out in the CRE endonuclease producing cell. So that's an example of how you would produce an adenovirus with just vector DNA in it. And that vector DNA is missing all the viral genes, so it can't replicate on its own. It can get into cells and deliver the transgene. It will be transcribed, but it will not propagate. Adenoviruses can be further modified in many interesting ways to make them even more useful, and some of those modifications are shown here. For example, you can modify the capsids of the, so sometimes when we say vectors, we're also meaning virus particles. I usually refer to a DNA, but sometimes people refer to a vector as a virus also. You can modify these viruses with microvesicles or various polymers that basically reduce their immunogenicity, so they're less likely to be cleared. And you can experiment with these in animals and then decide on the ones that you want to try out in people. You can also modify these viruses with various peptides or natural ligands on polymers. So we're decorating the virus capsid. For example, we have an FGF2 molecule here, fibroblast growth factor number 2, and that would target to cells that have a receptor for that particular fibroblast growth factor. So in other words, we can target these vectors to specific cell types if we know there's something on those cells that's specific, and we can modify the adenovirus to interact with them. You can also engineer the fiber protein itself. The fiber, of course, is what binds the cellular receptors. We can change the tropism of the vector by modifying the fiber, putting peptides, for example, on the fiber proteins. So we can do this in order to change the tropism or restrict the tropism so you don't have adenovirus infecting everywhere. And this is something you can do with many of the vectors that we're going to talk about. Another very popular gene delivery or gene therapy vector is adenovirus-associated virus. This is a member of the parvovirus family. To refresh your memory, these are rather small icosahedral particles. They contain a single-stranded DNA genome. The genome is diagrammed at the top right here. Single-stranded DNA with the unusual T hybridized ends at either ends, which serve as initiation sites for viral DNA replication. And these encode a handful of mRNAs of different proteins, including capsid proteins and proteins that are needed for DNA synthesis. This being a small virus, it has to recruit the cellular DNA replication machinery. A subset of the parvoviruses are helper-dependent. Last time we talked about helper-dependent viruses. This is one of them, and you can tell by the name, Adenovirus-associated virus. This needs adenovirus functions in order to replicate. And these are very popular for gene therapy because one of the main reasons is that they have long-term gene expression. When you infect a cell or an animal with these viruses, they persist for long periods of time. And so that's desirable if you want to give someone a single inoculation of vector and have them produce the product for a long period of time. There are also multiple serotypes available, and we can make new serotypes as well. So here in panel A is a diagram of the viral genome showing you the elements, the inverted terminal repeats, then the region encoding the replication, the rep proteins, and the capsid proteins. And the way you make vectors out of these is you delete most of the coding region of this virus. That's shown in panel B. And then you insert a transgene. So that DNA on its own will not be able to replicate because it doesn't have the rep protein in it. So what you do is you would transfect that DNA into cells along with two other important things. First, a separate plasmid encoding the rep and the cap genes to provide the capsid proteins and the proteins needed to initiate viral DNA replication. And then you have to provide the adenovirus functions. We know exactly what they are. So we can actually have plasmids. We don't have to infect these cells with adenovirus. We can have plasmids containing the right genes. In the case of adenovirus, it's E1, E2, E4, and the VA RNA. So we can have those on plasmids. We put them all into cells. What we get out is a recombinant adeno-associated virus. Only the vector is packaged, and the resulting preparation contains the gene that you want to give to patients. You can also modify these capsids extensively. Really amazing things have been done. So for example, we have on the left here different colored bars which represent the genomes of different adenovirus that give rise to different immune responses. So they're not cross-reactive. They're different serotypes, if you will. You can take those and do a variety of things to generate even more serotypes. You can mutagenize these capsids, just subject them to random mutagenesis so you have multiple amino acid changes within the capsid. You can shuffle the bits amongst the different capsid proteins, so you can see these recombinant capsids here. And then you can even insert other peptides throughout these sequences. And in the end, you put all these modified capsids into cells. You make virus capsids. Not all of them are going to make capsids, only the ones that are competent to do so. But you do this in mass, and then you can do a variety of experiments. You can infect cells and see which ones of these will bind antibodies. And then you can base what you proceed with ones that don't bind any of the known antibodies against the virus, for example. And you could take those that, if this column has antibodies to adeno-associated virus on it, it will pull out all of those that are still reactive to those antibodies. The ones that don't will pass through. You can subject them to another round of vector modifications. And of course, eventually you can put them in an animal to make sure that they're still able to replicate. You can make sure that they're still a vector. So this way you can make lots of new serotypes for using in people, because again, once you use one of these, if you want to go back again, you're going to have an immune response to contend with. Retroviruses are very popular gene therapy vectors. These, of course, are enveloped RNA viruses. They have plus-stranded RNA in them, and these viruses, of course, have reverse transcriptase and integration as an essential part of their replication cycles. And we have two different kinds of retroviruses that are used for gene therapy. We have retroviruses with simple genomes, like avian leukosis virus, RhCMV virus that we've talked about in this course. They encode a GAG, a POL, and an envelope gene. And we also use viruses like HIV-1 that have complex genomes. Besides the core proteins, gag, pol, and envelope. They also encode a number of other accessory proteins, smaller proteins that have a variety of functions that we've talked about. So both of these are used for vectors, and you may be thinking, why are we using HIV? This is a nasty virus. Well, of course, we modify it extensively so that we don't have any of the problems associated with infection. So many vectors are based on HIV, which is a lentivirus. HIV is interesting and useful as a vector because it can infect non-dividing cells. The retroviruses with simple genomes, they need a dividing cell to infect, and not every cell that you want to deliver a gene to is going to be dividing. So HIV can do that. And of course, HIV and retroviruses in general will integrate as a proviral DNA into cells, so you have long-term expression. You can put up to 8 kb, not as big as adenovirus, but other advantages in many cases will point to retroviruses. One of the negatives, of course, is the possibility for insertional mutagenesis. When a provirus integrates randomly into your genome, at some point there may be an oncogene next to where it is integrating, and that oncogene can be turned on inappropriately and lead to cancer. Now, to get around this, and we didn't know that, or we didn't appreciate the extent to which that was a problem until there was a therapy trial and some of the recipients developed tumors based on insertion. So now we modify the vectors to either inactivate the 3' LTR so that it cannot activate a neighboring oncogene, or we can make these viruses integration deficient, which of course takes away a lot of the utility, but gets around the problem. When we use these as vectors, we typically do what we call pseudotyping. What that means is we simply substitute the retroviral glycoprotein with that from another virus. And the VSV-G glycoprotein is often used. So the resulting particles would be HIV with a VSV glycoprotein. They're called pseudotypes. And the reason we do that is to expand the tropism, because if we use the HIV glycoprotein, it would restrict it to CD4 producing cells. The VSV-G glycoprotein can infect just about every cell out there. So if you want a broadly infecting vector, you use VSV-G. So here's how you make a retrovirus vector. You can actually make retrovirus particles just by putting 2 plasmids into a cell. You can take a plasmid encoding the gag-pol region. That's called the capsid plasmid. And the capsid is the protein on the interior. And then you can take another plasmid encoding the envelope glycoprotein. You put these 2 in cells, you will get an empty virus particle. There's no viral genome here. You could actually just put the capsid plasmid in by itself. It would make particles, but they wouldn't have envelopes, and so they would not be able to infect cells. If you want to put a foreign gene in here, you add a 3rd plasmid to this mix. You have your capsid plasmid, you have an envelope plasmid, and then you put in a plasmid with your transgene, and this, of course, is going to be flanked by LTRs so that it will eventually integrate. And now the 3 of these together will give you a retrovirus containing a foreign gene, and then that will be used to deliver genes to people. So, of course, envelope here, it can be substituted for the VSV-G to make pseudotype particles that have an expanded tropism. Vaccinia viruses have also been used as vectors for gene therapy. This is a virus we've talked about very briefly in this course. Big double-stranded DNA containing viruses. The pox virus is 130 to 375 kilobase pairs of double-stranded DNA, which is linked at the loops. So it's covalently linked. If you melted this, it would be a single-stranded circle. And of course, smallpox virus is a pox virus. That's the virus that we've eradicated, the only virus we've been able to eradicate. And a common vector derived from vaccinia virus, pox viruses, is called the modified vaccinia virus Ankara. That's the name of the strain, or MVA. You'll see that in some subsequent slides. So basically what was done there is someone took vaccinia virus, which was used as a smallpox vaccine for many, many years. They passaged it many, many times in chicken cells until it was well adapted to chicken cells and is no longer able to infect mammalian cells. There's an assembly block now as a consequence of that passage. This MVA was actually used towards the end of the smallpox eradication campaign. It was given to a number of people. It was effective at controlling smallpox. But now it is used as a vector because it has a number of properties. It's safe in people. We knew the original smallpox vaccine was safe, and this is derived from it. It's even safer. You can work on it in BSL-1 laboratories, the lowest containment possible, and so it's very easy to work with. It's an intrinsic adjuvant, and so the virus particle has lots of proteins in it which apparently stimulate immune responses, and it has large capacity, as you can see here, from the size of the genome. You can cut a lot out, still have lots of viral genes left, and you can add whatever you want. The way that you put genes into vaccinia virus is shown here. So you take the modified vaccinia virus Ankara, MVA, and you infect cells which have also been transfected with a plasmid containing whatever gene you want to substitute into the MVA genome. And what will happen is the transgene in the vector will recombine with the viral DNA, and you get out virus particles that have the DNA inserted. The reason that they do this is because you flank the transgene with sequences that are homologous to the viral genome. So you put sequences out at either end. These will recombine into the viral genome, and you can target it to a non-essential part of the genome, and then you get recombination, and then you simply screen the viruses that come out of these infections for the presence of your transgene. So it's pretty easy to make recombinants in MVA. So here's a summary of these viruses that we've talked about. There are others as well, but I would say these are the main ones. And you can see some of the advantages and disadvantages. You know, sometimes we have a reasonable capacity. The highest, of course, adenoviruses and the pox viruses. Some of the advantages, genome persistence is really important. Of course, non-pathogenicity is important as well. And then some disadvantages, immunogenicity, low capacity, oncogenic potential obviously is an issue, capsid stability, et cetera. So, you know, everyone works on different variants and different kinds of gene therapy vectors. And over the years, it takes many years to sort out the ones that work the best for whatever the particular application is. Just to give you an idea of what kinds of applications we are using gene therapy for, this is a summary of indications that are being addressed in clinical trials. So many of these vectors are now in clinical trials, and the number of trials is shown here by the N. So you can see there are quite a few clinical trials going on. And on the left is a pie chart showing you what kinds of indications these clinical trials are addressing. And you can see the main one, 64% cancer, the yellow part of the pie, 1,376 clinical trials. So using viral vectors for cancer therapeutics is really a big deal. Monogenic diseases, about 10% or so. So these are cases where there's a defect in a single gene leading to a disease, and you can easily fix it by giving people the normal gene. And we have infectious diseases, vaccination, of course, and a variety of other diseases, cardiovascular. We'll talk about some neurological and ocular diseases today, but you can see quite a few different varieties of approaches for gene therapy. And on the right is a pie chart showing you what vectors are being used in these gene therapy trials. You can see 22.5%, that's the biggest one, is adenovirus. And I think that's largely because it's been used the longest. I'm not sure it's always the best, but it's been around a long time. We have retroviruses. In many cases, naked or plasmid DNA is used. I talked about some applications of plasmid DNA vaccines last time. Vaccinia virus, 7%, adeno-associated virus, 5.8%, and a few others, including pox, herpes virus, and so forth. Anyway, so it's a broad range of things. Now, this slide shows you some of the general approaches we use to deliver viruses or viral genes to people. There are really two ways you can do this depending on the application. You can direct deliver or you can do cell-based delivery, all right? So in direct delivery, you make your viral vector. In this case, maybe you want to deliver a therapeutic gene. But we'll see in a bit that you can also do tumor therapy by this. You just inject or infuse the vector into the patient, and hopefully the virus will go through the right organ and produce the gene or produce whatever the effect is that you're looking for. As you might imagine, this is a very easy approach, but it's not always targeted. Your virus may not get to where it needs to go. If it's targeting the liver, I mean, when you put something in the blood, that's the first place the vector is going to go, so that's fine. But if you wanted to get a virus into the brain, this would be quite difficult. And sometimes you have to directly inject the virus exactly where you would like it. So for a brain tumor, you have to inject the virus into the brain. But we'll talk about some applications in a bit where that has worked, putting the virus into the blood. The other approach is very neat. And here what you do is you take your virus vector and then you infect cells in culture. And the cells you can take out of a patient. You could take out blood stem cells, for example. We've learned in recent years how to purify stem cells from the blood. So stem cell meaning it can be differentiated into almost any other cell type. You can culture those and then you can infect them with your virus, grow them up, and then put them back in the patient. And then those infected cells will home into various organs and propagate. Now, in the old days, we used to have to take bone marrow from patients in order to do this. That's a very difficult procedure. It's extremely painful, not a lot of success. But now we can take peripheral stem cells and do this. So this is a very nice approach because you do all the infection in the lab in a culture dish, and you can ensure that all of your cells are infected. Now, a couple of examples of gene therapy. First, looking at infectious diseases. Last time when we talked about ways to prevent AIDS, I mentioned this, and I want to just reiterate it because we went over it very briefly. Here, what was done is to identify from patients a broadly neutralizing antibody. That is, an antibody that will neutralize many, many different subtypes of HIV-1. And you can do this by taking B cells out of patients and taking single B cells which are producing antibodies, and you can sequence them to figure out exactly the antibody that's being produced, and then you can clone the gene for that antibody. So one of these highly broadly neutralizing HIV antibodies was cloned into an adeno-associated virus vector. So here's AAV, and remember we take out most of the coding region of AAV and we substitute it with our foreign gene. In this case, it is a construct designed to make a heavy and light chain of this broadly neutralizing antibody. So that's with IgG, heavy chain and light chain. Remember, antibodies are made of separate polypeptides, two heavy and two light chains. And in this vector, you make them both together. But look, this is just an example of why we do basic research on viruses. In the middle of the heavy and light chain coding region is a coding region for 2A. This is a protein we mentioned very briefly a long time ago. It's a Picornavirus protein that can shut off host translation. But in some Picornaviruses, it actually contains a ribosome stop signal for reasons that people don't quite understand. As the ribosomes are translating 2A, they pause briefly and then they start again, and you end up getting two separate polypeptides. So someone found this out a long time ago in a Picornavirus, and now it's turned out to be useful for vectors because you can put this sequence, which is very short, it's less than 10 amino acids, in an mRNA, and you will get two separate polypeptides from it. So that way you get the heavy and the light chain. All right, so you take this adeno-associated virus, which you would make using the approach I showed you earlier. You can infect mice with it. You infect humanized mice so that you can then infect them with HIV. They have the immune system of a human. Otherwise, a mouse immune system will not be infected by HIV. And then you can see if making this antibody, this broadly neutralizing antibody, will protect them. So you're not immunizing them with the protein, you're giving them, it's kind of a passive immunization, except we're giving them the gene for the antibody, so it's going to last a long time. And in fact, these mice make grams of antibody for their entire lives, so it's long-term production. And on the right, these mice have been challenged with HIV. So if you look at the bottom panel, percent uninfected. So the immunized mice are in red. They remain uninfected. And this is, every week they challenge them with HIV. So here we have 21 challenges. Every week you get an injection of virus. And these mice that make these broadly neutralizing antibodies never get infected. And the mice that get a vector control, of course, they get infected. And on the top is the number of viral copies per mil of plasma. And again, the red lines are the mice that get this broadly neutralizing antibody. They're fully protected, whereas the cohort of control immunized animals get infected. So this is a promising approach that is going into a phase 1 trial in people, which means to assess its safety. You give it to 10 or 20 people, and you make sure that they're okay. And presumably making a lot of antibody won't make them ill. Another example of a vaccine using an interesting technology is an Ebolavirus glycoprotein vaccine in a dual kind of immunization using both adenovirus and MVA, vaccinia virus. And this is a series of experiments where they're either giving animals, and these are non-human primates, chimpanzee adenovirus. So the idea would be here, if this made it into people, they would not have antibodies to adenovirus. Adenoviruses commonly infect all of us. So you use a chimp adenovirus, which is not likely to have infected you. And so they clone the glycoprotein gene for Ebolavirus into a chimpanzee adenovirus type 3. 10 to the 11th PFU protects only 50%. You can see if you go down tenfold in the dose, you don't get any protection. So that's not very good. Then they tried a prime boost strategy, which they give you first chimpanzee adenovirus with the glycoprotein, and then they go back with something else. So if you do add, which is kind of like a traditional prime boost, you protect 33% of the animals. Not much better if you use a different serotype of adenovirus, the idea being maybe the second one didn't do very well because of an immune response, but that's not it. However, if you do adenovirus followed by MVA, vaccinia virus producing the glycoprotein, you get 100% protection. And this is a protection against Ebolavirus disease by intramuscular challenge. So that's promising. Unfortunately, right now, there is no Ebolavirus disease globally, so none of these Ebola vaccines can be tested. Here's another Ebola virus vaccine, which uses a different strategy, which I think we mentioned. We take the genome of vesicular stomatitis virus. This is a pathogen of cattle, does not cause serious infection of humans. You can replace the glycoprotein gene of VSV with the glycoprotein gene of Ebola virus. You make a pseudotyped virus. It looks like VSV, but on the surface, it's Ebola virus. This has been tested in non-human primates as well. This is percent survival. All right, we have animals that were either immunized with that recombinant VSV or just VSV alone. And then we challenged them intranasally with 1,000 PFU of real Ebola virus. And then this is the course after 28 days. So in the vector, the control animals, they're all dead within 6 or 7 days or so. And the vaccinated animals are all protected. So this is promising as well. If it can go into people, we might have ourselves several Ebola virus vaccines. And the last example I want to show you is an example we talked about the other day. This is another kind of HIV vaccine, and this is using cytomegalovirus as a vector. You put the envelope glycoprotein of HIV into a cytomegalovirus, the idea being that these are persistent viruses in humans. Once infected, you remain infected for your life. And so it might be good to be making, this time we're making the glycoprotein, not an antibody, but the glycoprotein itself. And since this experiment was done in non-human primates, rhesus macaques, we use a rhesus CMV, or with an SIV glycoprotein, and then we challenged these animals with SIV, because again, you can't use HIV in non-human primates. And what you can see here is that we're looking at plasma viral load on the y-axis with time post-infection, and all the blue lines, these were animals that got just vector. They did not get immunized with viral glycoprotein, so they're challenged. They all get infected with SIV. And then on the bottom are the animals, different colors here. You can see they're listed RH with different numbers. These animals received the envelope glycoprotein in the rhesus CMV vector. And you can see, with the exception of a few blips early on, most of these animals were protected. There was one animal in red here that clearly, after 28 weeks, started to produce virus. So why that's happening has to be investigated. But there's a lot of excitement over this approach for eventually going into people because of the persistence of the vector. Let's talk a little bit about monogenic diseases and how we can approach those using gene therapy. These are diseases caused by mutation in one gene, as you can tell by the name. There are over 6,000 of these, and they amount to 1 in every 200 live births globally. So a lot of people get born with a problem in one of their genes, and often it's a serious business. Some of these individuals die very quickly, others die after 5 or 10 years or so, or have lifelong problems. So you would like to give them a normal copy of the gene. So this is a perfect candidate for gene therapy, and there are over 1,800 clinical trials going on right now to try and correct some of these monogenic diseases. And this is just obviously a partial list of some monogenic diseases that are amenable to gene therapy. Here is the name of the disease on the left. What the defect is, we know in these cases, of course, the specific gene and the mutation that causes the disease. Here's the incidence here. So some of these are low frequency, some of them are more common than others, and what vectors are being used in some of the clinical trials. And we're going to talk about some retinal diseases, some congenital blindness diseases that are being studied using Adenovirus-associated vectors. I'll mention X-linked adrenoleukodystrophy, but you can see there are many others, blood disorders and others as well. Of course, there are thousands more not listed on this slide, but often the ones that are used initially have a good incidence, and we know exactly what's going on with the defect. Now, the first viral gene therapy in humans was in 1993, and this was for cystic fibrosis. And cystic fibrosis, the individuals have mutations in the cystic fibrosis transmembrane conductance regulator gene, CFTR. And this particular individual was a 23-year-old male who was homozygous for a particular mutation, which made ion imbalance in his lungs improper, and these patients, of course, get lots of infections as a consequence. And what they did was to deliver to this patient one of the earlier generation adenovirus vectors, E1, E3 deleted, with the transmembrane conductance regulator DNA integrated into the viral vector. They gave him 2 × 10⁸ PFU by inserting a tube, and you can see they took a picture of this going on here. They put a tube down into the lungs, and then they spray or they aerosolize the virus down in the lung. And you can see the doc here is watching himself on the monitor there, making sure he's going to the right place. And then they measured the production of the CFTR protein in his wash. You can see that's shown on the graph here. This is exogenous over endogenous. So he makes a certain level of CFTR, but of course it's mutated. And this is a ratio of the adenovirus encoded protein over the endogenous. So you can see the initial inoculation, he's making 100-fold more than his endogenous, and then it goes back down very quickly. They gave him a second installation, got a little bit less production there, and that's probably because of the antibody response to the primary. And then by the third time, no response whatsoever. So by then he had great immunity to this vector and didn't respond. So this was the first one done in people. This was a historic occasion. We learned a lot of things, like we have to change the serotype after each installation. But these are transient vectors. They don't permanently cure whatever problem is. And so to do that, you have to use a vector that persists. Now, after a number of gene therapy trials, there was one serious setback, which of course is Jesse Gelsinger. He was the first person to die in a gene therapy trial. He was an 18-year-old, pretty healthy kid who happened to have ornithine transcarbamylase deficiency. But he was okay, but he volunteered for this trial just because he wanted to do a good thing. And what they did was give him an adenovirus vector. They injected him with the normal OTC gene. This was done at UPenn. The idea was, this is an early trial to see how he fared and how much gene expression there was and so forth. But unfortunately, 4 days after receiving the vector, he died. He had a massive immune response against the vector. He had multiple organ failure. And of course, this put a damper on gene therapy for a while. It turns out that many things were wrong with this trial. Unfortunately, rules of conduct at Penn were broken. They probably gave him way too much vector, and a lot of things which, unfortunately, in retrospect, should have been done differently. If you just Google his name, you'll find a very moving essay by his father about what went on here and so forth. But eventually, the trials resumed. One of them was this trial for X-linked severe combined immune deficiency. In this case, there were 2 trials in London and Paris with infants with deficiencies in IL-2 gamma receptor gene, and they gave them a retrovirus with a normal copy of this gene. What they did is to take out bone marrow hematopoietic precursors. They cultured them, they infected them with the retrovirus, and then infused them back into the patients. 4 out of 9 patients in Paris, these are infants, developed T-cell leukemia 3 to 6 years later, and 1 in London. Now, so this trial actually worked. The treatment worked. It had restored function of this gene, but unfortunately, the vector had integrated next to an oncogene, because remember, integration is random. And so after multiple years, this integration led to integration next to an oncogene, and that turned on the oncogene. That leads to cancer. The cells get transformed, they start to grow uncontrollably, and eventually they sustain enough mutations to form a tumor. And so at the time, there were 27 ongoing trials using retroviral vectors. They all stopped. They said, nope, no more. You got to stop, figure out how to deal with this. And as I said, they came up with ways to inactivate the 3' LTR or prevent integration. But that's the way it works when you do cutting-edge things and you're involving clinical trials. Sometimes things go wrong. One disease that in which gene therapy has worked is called X-linked adrenoleukodystrophy. This is a CNS problem where patients have a defect in a transporter called ABCD1. And what they did in this study was to take bone marrow, again, stem cells, culture them in vitro, and then infect them with a lentiviral vector that was producing a normal ABCD1 transporter gene. They would grow these cells in culture, make sure the gene's being expressed, then put them back into patients. And this was a small trial of a few individuals, but in this disease, as the kids age, they degenerate neurologically, and their status had stabilized or improved. And so these were CAT scans of the brain showing on the right the extensive pathology in an advanced case, and on the left, one of the kids with the infusion of this gene. Here's another great example of gene therapy for inherited retinopathies. All right, these are very common conditions that lead to blindness. So far, they're not treatable. And they are monogenic diseases. They have a mutation in one gene, and the genes involved are either retinal photoreceptors or retinal pigment epithelium. You can see on this diagram of the retina here. Of course, there are a large number of cell types making up the retina. The retinal pigment photoreceptors are up here. The pigmental cells are down here, and a variety of other cells. And there are specific mutations in each of these different cell types that lead to these blinding conditions. And on this table is the remarkable number of different trials that are ongoing using 3 different vectors: adeno-associated virus, lentiviral, and adenovirus vectors. and each of them containing genes from different cells in the retina. So the RPEs, the pigment cells and so forth. Each of those, there's a different gene. Of course, these are individual trials where in one trial they're giving this particular gene with AAV and so forth. You can see a lot of study has gone into this, knowing which genes are defective, and so you can put the normal counterparts in the vectors and give them to patients. The way these are done is diagrammed on the left here. You actually have to inject the virus right into the retina. So you have to put the needle through the eyeball, right into the retina there. You have to have a steady hand, obviously. In this case, we're putting adeno-associated virus in there. And there has been some success with these. In one particular study, they were looking at one gene called RPE65. That's a protein needed for photoreceptor function in dogs. They showed that a single injection, subretinal injection, as I showed you, of the vector with the canine gene restores visual function in these dogs. And in people, it's been to both phase 1 and 2 trials, and for 2 years, they've had visual improvement after giving these AAV vectors. So that's one of the nice things about the AAV vector. You can have sustained production of the transgene and the potential for a long visual improvement. One of the people who has worked a lot on these vectors is Catherine High. She's a doctor at UPenn, and I interviewed her for an episode of TWIV, and she is quite knowledgeable about all the history and what goes into this. So if you're interested in more, check that out. So some of the successes for viral gene therapy, just a few. SCID, adenosine deaminase deficiency, the Leber amaurosis, which I just told you about. Hemophilia is another one, thalassemia, and a fat metabolism disorder caused by a defect in lipoprotein lipase. So these just get more and more numerous. This works, there's no doubt about it. It's just a matter of time when it becomes routine. Let's talk about viral oncotherapy now. This is killing tumors with viruses. We have a number of different approaches that are really interesting. Sometimes animal viruses selectively replicate in human tumors. You just get a virus from an animal, for whatever reason, it grows really well in human tumors, but not other human cells, so that's perfect. Sometimes we modify the virus to target and kill tumors with immune enhancement, as we'll see. And I made a video last year with Linda Coughlin, who's from Ireland, and she works on this sort of approach, and she gave an 8-minute version of how this works. So often we target viruses to tumors by receptors, for example. We can alter viral glycoproteins like the measles virus HA to target tumor cell markers. We've engineered herpes simplex virus glycoprotein to target a particular protein, or sometimes we can put antibodies on the surface to target proteins that are on tumors. We can modify adenovirus, as I showed you earlier, to include domains that recognize tumor antigens, and this involves modifying the hexon, which makes up most of the capsid, or the fiber, of course. We can retarget them. That's an important part of some of these vectors. And here, so that would be targeting the surface, the entry step, but you also want to target post-entry, and here are 2 ways you can do that. You can have what's called positive targeting. You use a promoter that is driving a gene that's going to kill the tumor. You use a promoter that only works in a tumor cell. And this, of course, depends on you studying transcription in a variety of cell types. And people have done this over the years as part of fundamental research. So here on the top left, we have a promoter that's not working in a healthy cell. So if you give this to a patient, whatever the toxic product is not going to be made in healthy cells. But the promoter works in a tumor cell, and there your mRNA will be produced, and you'll have whatever the toxic product is that you've encoded. Another approach, so that's called positive targeting. Another approach is negative targeting. Here, we incorporate into the vector genome the targets for microRNAs. All right, so a microRNA target will be recognized by a short interfering RNA, degrading the viral genome and inhibiting replication. And so here, for example, you can choose a microRNA that you know to be present in normal cells and insert the target of that microRNA into your vector genome. And so when the virus infects a normal cell, it'll be degraded by the microRNA system. On the right is a tumor cell, which doesn't have that particular microRNA in it. Your vector is unimpeded and it can replicate and target those cells. Now, it's very easy to find microRNAs. There are thousands of them, and you just do screens and you can find them that are specific to cell types. So on the right is a northern blot for two particular microRNAs, LET7A and miR-124A. And you can see miR-124A is only present in two different cell types that are shown here. The LET7A is in many different cell types, but it's not in this particular cancer cell, Ntera2. So you can pick a microRNA that's not in a cancer cell, and that way your vector will be unimpeded, because remember, the vector, you've built in the target for the microRNA. We also arm vectors in ways. Besides targeting them, we put genes in them that do a variety of things to enhance killing. Now, it's very hard to infect 100% of the cells in a tumor. There's just no way you're going to do that. Even if you inject the virus directly into the tumor, it's not going to infect 100% of the cells. So what you want to do is put genes in the virus that, when produced, will help kill the surrounding uninfected cells. All right, so this is called bystander killing. The cells that are not infected will be killed as well. And we put a variety of products into these vectors. We'll talk about some of them. Prodrug convertases, if you remember acyclovir and TK, that's going to bring back memories. Ion transport proteins and immunostimulatory factors. We also use vectors that infect animals, and normally not people, but for some reason they infect tumors. And one of them is Myxoma virus. This is the same virus that was introduced in Australia to kill the imported European rabbits. It doesn't replicate in any non-rabbit host, but it infects many different human cancer cells. How would you know this if you didn't try? It's another plug for just doing basic research and following your curiosity. This is probably because the cells don't make an antiviral response, and pathways related to transformation growing incessantly are induced, and that lets the virus grow very well there. So these Myxoma viruses are being used to treat a whole variety of human tumor types, different tumor types here. You can see the cancers of different sorts on the left, acute myeloid leukemia, pancreatic cancers, gliomas, and these are all done in mice, where they transplant cancers into mice, and then, so you can see tumor establishment here, they're transplanting them into mice, and these are immunosuppressed mice, so they won't reject the tumor. And then you put the Myxoma virus in a variety of ways into these animals, and you can see many of them have had remission of the tumor or destruction of the tumor. So again, Myxoma virus shows a lot of promise for treatment of a variety of human tumors. These haven't gone into advanced phases yet, but I believe they will. A Picornavirus called Seneca Valley virus is being used to treat a variety of cancers. This was initially identified as a contaminant in cell culture medium, and it turned out it was from either the serum from the cow or trypsin from pigs that was used to split the cells. So this is an animal virus that was just stumbled on, and it turned out to have selected tropism for a variety of cancers with neuroendocrine features, as you can see here. In mouse models, this has been very good at eliminating these tumors, and it's now in a phase 2 ongoing clinical trials in humans. And again, it's an accidental discovery. Someone discovered this and characterized it and then found that it infected tumors, and there it goes. Measles virus is another one that's been used for anti-tumors. The vaccine strain is already attenuated. This is the vaccine strain that we use to immunize against measles virus. It turns out that it preferentially replicates in tumors. This was not something that was selected for when the vaccine was being made, but it's something someone found out. It replicates in tumors probably because the virus can't antagonize antiviral proteins of the host cell. And what they did with this vector is they included a gene for a human sodium iodide symporter. This is a membrane protein that can transport ions across the membrane. And what that does is when you give this to people with tumors to treat their tumors, you can give them gamma-emitting isotopes, and you can visualize where the virus is infecting because of this symporter molecule. And then if you give beta-emitting isotopes, they get taken up preferentially into the cells with this NIS symporter, and it's a way of focusing the killing on those cells. So here is one clinical result of measles virus therapy. These were 2 patients with multiple myeloma. These are tumors of B cells, of course, which are the ones that make antibodies in you. And these are refractory to treatment. Most people with multiple myeloma eventually are going to die. And what they did was to give these individuals 10 to the 11th particles of measles virus intravenously. And one of the 2 had complete remission of her multiple myeloma. So these people have not only tumor cells floating around in their blood, but they lodge in different places and cause massive masses. And you can see on the top here, these are the 2 patients, patient 1 and patient 2. One of the characteristics of multiple myeloma is clonal plasmacytoma cells. So from this FACS plot, you can see pre-treatment, this person had a lot of clonal PCs. After treatment, they were all gone, normal PCs. And the second patient as well. Pre-treatment, lots of clonal PCs. After treatment, none. And in one of the patients, she came into the trial with a lump on, I think a 3 centimeter lump on her forehead, which you can see here in this imaging. And after treatment, it's gone. So the virus went into these tumors and killed the cells. And this is a remarkable achievement. Herpes virus is also used for certain kinds of tumor therapy. This one is called Telimogene laherparipec. I don't know why they would call it something, but anyway, they call it TVEC. And this includes the genes for granulocyte macrophage colony stimulating factor. This is a protein that stimulates the production of these cell types, and basically when the virus infects tumor cells, it makes this product, and it will attract other immune cells into the area to further kill the tumor. The virus has been deleted for 2 genes, which make it tumor specific. And in addition, the ICP47 gene has been deleted. ICP47 is one of those viral proteins that inhibits antigen presentation. So these cells will not be recognized by T cells as virus infected. They will not be eliminated. After a phase 3 for melanoma, this gave a good response, and it's now been licensed. Here's the FDA, or the newspaper announcement in Forbes. It was approved last year. And this article has a pretty good summary. It's injected directly into melanomas. It's a version of herpes that's been genetically modified, so it only replicates in cancer cells. Also encodes GM-CSF to recruit immune-boosting cells, and that's how it works. The only quibble I have is they call this a modified herpes bug. Okay, if you ever write for Forbes, could you not use bug? Right? It's a virus. What is wrong with saying virus? Did people not understand the word virus? Otherwise, it's pretty well written. So that's been approved. Here's another cool one, Adenovirus CG0070. This virus, armed with GM-CSF, preferentially replicates in RB-deficient tumors. This is in advanced trials for bladder cancer. They inject the virus right into the bladder. That's what intravesical means. Look at this, how this works. This is brilliant. You remember, there's a cascade of gene expression with adenovirus. Immediate early, and late expression. And nothing happens until E1A is made. And E1A, the little purple eggplant or kidney here, what that has to do is bind RB to free up the E2F proteins that the virus needs for early transcription. All right, so binding of E1A to RB frees up E2F. E2F can then go on and allow transcription of subsequent genes. So what they did is build a virus, which is shown on the bottom here, where it will only replicate in cells that lack RB, and that's typically tumors. How brilliant is that? So what do? They took the left end, here's the E1A gene. They took out the normal promoter for the E1A gene, which will work in pretty much any cell, and they substituted the E2F promoter to drive E1A. Normally, E2F is bound up to RB, so this promoter won't work in a normal cell, and there's no E1A to bind RB in a normal cell. There is no E1A produced here to free up E2F. So what will happen is this will get into a tumor that lacks RB, where E2F is free to begin with. E2F will bind the E2F promoter, cause production of E1A, and it will start replicating. And then downstream here, they put in a GM-CSF gene under the control of a late promoter, which is activated by E1A. So E1A will only be made in RB-deficient tumors because the E2F promoter that drives E1A will not work unless the RB is not present. Because in a normal cell, RB will be bound to E2F, and this virus will not replicate. So it leverages everything we've learned about transformation, RB, E1A, and makes it a virus specific for RB-deleted tumors. Another adenovirus called Ankara has been licensed in China, and here the E1B55K gene has been deleted, and that is needed to degrade P53. We talked about this a while ago. P53, remember, will kill the cell if it senses viral DNA replication in it, but the E1B protein will degrade P53, and so they took out E1B, so the only cells this virus will replicate in lack P53, and many tumors lack P53. This virus will not grow in other cells, but it will grow in E1B deficient cells, tumors. Brilliant application of molecular biology. Now here we have a modified vaccinia virus. I love the name of this company, Generex. Isn't that great? A little tip of the hat to Edward Jenner, who made the first vaccine. Generex JX594, they put GM-CSF in it. They deleted the TK gene. This happens to be a gene elevated in tumors, so that makes the virus more specific. And what they did in the trial is to inject this intravenously. They want to see if this virus can reach tumors that are all over the place and replicate in them. And they did this study in 23 patients with advanced treatment refractory solid tumors of a variety of types. And this virus was able to get to these tumors and replicate. And you can see here in this immunofluorescence, we're looking for viral proteins. The cancer tissues, so we have rectal, endometrial, and colon tissues, normal in cancer, normal in cancer. You can see the normal tissues are not infected with the virus, which is the green color. The cancer tissues are all infected. And half of the patients, the virus replicate, and they had anti-tumor activity. So the idea, again, is you put the virus intravenously, it can reach a variety of tumors, and it's specific for the tumors. So it doesn't infect other cell types because of the modifications. We also put prodrug convertases in some of these viruses. For example, thymidine kinase, remember, which converts a prodrug to a phosphorylated drug. Cytosine deaminase is a similar approach with a different prodrug. And what happens here, you have a tumor cell which is infected by your vector. The prodrug convertase, you then treat these people with the prodrug, 5-FU or ganciclovir. Then the prodrug is converted to an active drug in the cell, and it can diffuse to neighboring cells and kill those as well. And this is a virus that's under testing for that. This is called TOCA 511. It's a retrovirus which has cytosine deaminase. You give this intratumorally or intravenously to patients along with 5-fluorocytosine. And again, the virus gets to the tumor, and then the 5-FU or the cytosine gets into the tumor as well, is converted to a toxic drug which inhibits DNA replication, and that inhibits the tumor formation. So this is now in phase 2 for gliomas. Here we're putting the virus directly into the tumor. Even poliovirus has been modified for tumor treatment. This is the Sabin polio strain. The iris, the internal ribosome entry site, has been substituted with that of rhinovirus. This attenuates the neurovirulence of the virus. It's been tested in phase 1 for glioma, injected directly into the brain of glioma patients. This was featured on 60 Minutes because at least one of these patients has done well. Retrovirus, this is one of those viruses that is not pathogenic for humans. These come from a variety of animals. You simply take them unmodified. They will replicate in human tumors. And it's been a phase 3 trial for head and neck tumors as well as a variety of other studies as well. So those are just a few examples of how we can use vectors to do a variety of things, take care of tumors, repair genes, vaccinate and so forth. This emphasizes the importance of basic research. These therapies are possible because of advances in virology, recombinant DNA, and clinical science. And that's basic science, which we have to balance with translational science. Translational science is making vectors and putting them into people, which is obviously important, but you can't get there without the basic science. And right now, NIH funding is tipping towards translational science, and if we do that, we're not going to have the fundamentals to get there in the future. That was fast. That was a whole semester, so that's you at the first day of class. I want to thank you for taking this course. Just a few more announcements before we end. There is a survey at CourseWorks. Please fill it out. Let us know. If you like the course, that's good. Then I can teach it next year. So please tell us if you like it. If there's something you don't like, tell us. You have this week to finish the quiz for this week. I have office hours this week. And if you can't make that, you can email me and set up an appointment. But most of all, don't forget what I've taught you. I know as soon as you finish this course, a lot of these things are going to go out of your head. But look, you have all your videos. They'll always be there at YouTube. If you ever see something about a virus you don't understand, just look it up and try and figure out from what you've learned. It's all there. Maybe more than anything, you now know where to look for things. So as you go forth in your variety of careers, just remember virology. So I'll leave you with this last slide again, which I showed you at the beginning. I'm passionate about viruses, and I love teaching this course. I have loved telling you everything I know. And of course, I have viruses on my car, which emphasizes that. I want you to be curious for the rest of your careers. Don't assume that just because you don't understand something, you can't. Try and figure it out. And I would like you to follow me somewhere for the next years. I do a lot of podcasts. These lectures are all on YouTube. Every year I have a new set of lectures. This is my blog. This is my site for my podcast. I'm on Twitter, YouTube, and Facebook, and Instagram. And I use those to promulgate, to post information about viruses. So follow me somewhere, and please be curious. Thanks very much.